Hi everyone. Now let's talk about fixed dose combination FDCs. FDCs contain two or more active ingredients in single dosage forms. The challenge: each API can have different solubility, different release kinetics, different stability, different acid sensitivity. So, one method must differentiate each drug, quantify each accurately, interpret release profile correctly, all in the same test. For FDCs, dissolution is not just about release, it's about separating, measuring, and justifying each API in line with regulatory expectation. One tablet, multiple drugs, one scientifically robust method. In FDC's product, each API behaves differently. For example, one drug may dissolve very fast, another may dissolve slowly. Why? Because APIs can have different solubility, different pK values, different optimal pH conditions. Some drugs dissolve better at low pH, others need high pH to go into solution. Additional challenges: UV or PDA interference between APIs, drug-drug and drug excipient interactions, need to quantify multiple actives at the same time, different release mechanism in one tablet. For example, IR plus ER combination. That's why FTC dissolution method must be carefully designed, selective and well validated. FDC design directly drives the dissolution strategy. For example, blended tablets. APIs are uniformly mixed. One common profile but separate quantification needed. Layered tablet, bilayer or trilayer. Stage specific sampling. To track each layer's release, multi-particulate. Different coated beads for each API. Method must discriminate each fraction. IR and ER combinations need early and late time points to capture both phases. Sprinkle capsule. Mixed pellets must ensure uniform exposure and sampling. So remember, dosage form design decide the dissolution approach. One product, multiple release behaviors, one smart tailored method. For FDC products, independent solubility screening is critical. Each API must be evaluated separately for solubility across pH 1 to 8, pK and ionization behavior, log P and hydrophobicity, stability in selected media, possible excipients incompatibilities. For example, if API A dissolves well in acidic pH but API V does not, you may need a compromise medium or a two-stage dissolution method. One size fits all does not work for FDCs. A scientifically justified medium ensures both API remain soluble, profile are meaningful, data is regulatory ready. FDC dissolution is always a balancing act. Sometimes one medium over dissolves API A but under dissolves API B. So what can we do? Options include multi-stage dissolutions, adjusting buffer strength or pH. Key principles: choose a pH that dissolves all APIs adequately, or use stage-wise pH changes. Avoid surfactants that disproportionately boost. Only one hydrophobic API always ensures sink condition for the slowest dissolving API. For deeper insight, consider biorelevant media to understand in vivo behavior. The goal is one method that is fair to all APIs, scientifically justified, and regulator ready. Let me give you a practical example. An FDC contains API A solvent in acidic pH, API B dissolve in intestinal pH. In this case, a two-stage dissolution method becomes essential. Stage 1: pH 1.2 to evaluate API A release, and stage 2 pH 6.8 to capture API B release. This approach is essentially important for API requiring different pH environments, IR and ER combinations in one dosage form, entry coated plus immediate release components. Pellets with different coating functions, one medium cannot tell the full story. Multi-stage testing ensures each API is properly evaluated in its relevant environment. Right pH, right stage, right science. For FDCs with multi-particulate or coated beads, USP apparatus 4 is often better choice. Why? Because it provides controlled continuous flow, uniform exposure of all particles. In contrast, USP2 may cause conning, beads can accumulate at the bottom. Operator selection guide. USP2 most common starting points. USP1 when capsules or pellets float. USP4 best for very different API solubilities. Best for ER beads and complex systems. Always consider particle movement, conning behavior, uniform exposure, right apparatus, meaningful data. In FDCs, the faster dissolving API is often more sensitive to hydrodynamics. That's why RPM selection is critical. Your RPM must prevent conning, ensure uniform particle distribution, but be careful. Over agitation masks real formulation differences. Typical working range 50 to 75 RPM. Best practices: the test 2 or more RPMs. Use intentionally varied batches. Confirm the method is truly discriminatory. Choose an RPM that shows differences, not one that hides them. In FDC dissolution, UV spectra often overlap between APIs. This makes direct UV quantification unreliable. So, in many cases, HPLC or UPLC is required to achieve proper specificity and separation. Key considerations: careful wavelength selection if UV is used. Validate specificity for each API. Check carryover between injections. Ensure one sampling time suits for all APIs. Confirm API stability during analysis. Remember, if you can't separate it, you can't quantify it accurately. Good chromatography, credible dissolution data. In FTCs, sampling points must be synchronized. If the IR component finishes in 15 minutes and ER components last in 8 hours. Your method must capture both extremes. Typical strategy: IR APIs 5, 10, 15, 20 and 30 minutes and ER APIs 1, 2, 4, 6, 8, 12 hours. Key rules: use common time points for all APIs. Capture fastest and slowest release. Design profiles suitable for F2 compression. When applicable, one timeline must tell both stories. That's the art of FDC's dissolution. Regulators, especially FDA, expect dissolution method that can detect meaningful product changes. For FDCs, this is even more critical because of their added complexity. Your method must be able to detect changes in API ratio, coating thickness or uniformity, compression force, granulation process. Be careful. Overlay aggressive media or high RPM can mask real differences, creates regulatory risk. Best practices: test intentionally varied batches. Confirm your method is truly discriminatory. A good FDC method should reveal problems, not hide them. Discrimination, regulatory confidence. Validation for FDCs is more involved because each API must independently meet validation requirements. Validate accuracy and precision for each API. Confirms filter compatibility for all APIs. ensure stability of each API in media, demonstrate robustness across media prep, RPM, and PM. FDC dissolution method development is all about balance. You are managing the conflicting needs of multiple APIs in one product. A structured approach makes it successful. Understand each API, solubility, PK, and release behavior. Select a medium and apparatus that works for all API, not just one. Optimize hydrodynamics and sampling to capture multiple release profiles. Use specific analytical method. SPLC is preferred for accuracy. Validate each API independently and collectively. When science drives your method, you get accurate data, robust performance, and strong regulatory confidence. Thank you for watching. In next video, we will discuss pediatric dosage forms, tailored dissolution approaches. Please subscribe us for getting update on upcoming industry application oriented video and stay tuned because in this 62 video series we will cover dissolution method development, validation, troubleshooting and many real industry example. Please share your queries in comment section. See you in next video. Thanks.